Have you ever wondered if it's possible to create an exact genetic copy of a living organism? You're not alone. This exact question has intrigued scientists for decades and led to the development of what we now know as cloning. Cloning, in its most basic form, is the process of producing individuals with identical genes. It's like taking a genetic photocopy of an organism. This fascinating area of science has been the subject of countless movies and books, often with a futuristic or dystopian twist. But the reality of cloning is far from fiction. It's a very real and very intricate scientific process that has the potential to revolutionize the way we understand life itself. From Dolly the sheep, the first mammal cloned from an adult cell, to potential applications in medicine and biodiversity, the world of cloning is a labyrinth of possibilities waiting to be explored. Join us as we unravel the fascinating process of cloning. To fully grasp cloning, we need to understand DNA, the genetic blueprint of life. DNA, short for deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. It's a bit like an instruction manual, containing the information needed for an organism to develop, survive, and reproduce. Each cell in an organism's body has the same DNA, and most of the DNA is located in the cell nucleus. It is made up of two long strands arranged in a double helix. These strands are composed of 4 chemical units, or bases, known as adenine, guanine, cytosine, and thymine. The sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. In essence, DNA is the starting point for the magic of cloning. It's the blueprint that guides the growth and development of all living organisms, and manipulating it allows us to create genetic duplicates, or clones. One of the primary methods of cloning is somatic cell nuclear transfer, or SCNT for short. This technique may sound complicated, but let's break it down step by step to understand how it works. First off, we start with cell collection. Scientists collect somatic cells, which are essentially any cell in the body that isn't a sperm or egg cell, from the organism they wish to clone. These cells can come from various tissues, like skin or muscle. Next comes the nucleus extraction. The nucleus is the part of the cell that houses the genetic material. Scientists extract the nucleus from the collected somatic cell using specialized techniques. At the same time, an egg cell from the same species is prepared. The nucleus of this egg cell is removed, leaving behind an enucleated egg cell. This is an egg cell without a nucleus and therefore without any genetic material. Now we move on to nucleus transfer. The extracted nucleus from the somatic cell is then inserted into the enucleated egg cell. The result is a combination of the donor organism's genetic material with the egg cell. Once the nucleus transfer is complete, the next step is stimulation and development. Through chemical or electrical stimulation, the reconstructed embryo is prompted to begin dividing and developing. This process mimics what happens during fertilization and embryonic development. Finally, we come to the implantation stage. Once the embryo reaches a suitable stage of development, it is implanted into the uterus of a surrogate mother. Here it continues to grow and develop, eventually forming a clone of the original organism from which the somatic cell was taken. This entire process, from cell collection to implantation, is a delicate and intricate dance of science that allows us to create an exact genetic replica of an organism. It's a testament to the incredible advancements we've made in the field of genetics, and that, in a nutshell, is how cloning is achieved. It's a fascinating world of science where we're pushing the boundaries of what we know and understand about life and its creation. As groundbreaking as cloning is, it's not without its ethical implications. This technology, which has the potential to revolutionize medicine and biology, also opens a Pandora's box of ethical questions and societal concerns that need to be carefully considered. One of the most pressing ethical issues centers around the treatment of cloned animals. Cloning processes often result in high failure rates and abnormal development, leading to suffering and premature death among cloned animals. This raises questions about the morality of causing potential harm to animals for scientific purposes. The potential for human cloning also stirs intense debate. While some argue that it could have medical benefits, such as creating genetically identical organs for transplants, others warn of the potential for abuse. There are fears that human cloning could be used unethically, to create designer babies or to clone individuals without their consent. Can we, as a society, regulate this technology effectively to prevent misuse? Moreover, there's the issue of biodiversity. Cloning can potentially lead to a lack of genetic diversity, which is essential for the survival and adaptability of species. If we rely too heavily on cloning, could we inadvertently weaken the gene pool, making species more susceptible to diseases and less able to adapt to environmental changes? Then there's the philosophical quandary. Clones raise questions about identity and individuality. If a clone has the exact same genetic material as another being, what does this mean for its sense of self and its place in the world? Are clones merely copies, or do they have their own unique identities? These are just a few of the ethical considerations that arise with cloning. It's clear that while the scientific potential is enormous, the ethical challenges are equally significant. As we move forward with this technology, we must tread carefully, considering not just the scientific implications, but the ethical ones as well. These ethical considerations are as important to understand as the cloning process itself. As we delve deeper into the world of cloning, we must ensure that our scientific advancements are guided by a strong ethical compass, ensuring that we use this powerful tool responsibly and with great care. As we wrap up our journey into the world of cloning, it's clear that this scientific frontier is a double-edged sword. We've delved into the DNA, the blueprint of life, and understood the intricate process of cloning through somatic cell nuclear transfer. We've seen how cloning can replicate life down to the last gene, creating identical copies from a single organism. But with this great power comes great responsibility. We've also touched on the ethical dilemmas that cloning presents, from the treatment of cloned animals to the potential for human cloning. It's a complex landscape that requires careful navigation. Yet the potential of cloning technology is undeniably vast. From medical advancements to understanding genetic diseases, cloning could revolutionize our world. As we continue to push the boundaries of science, let's do so with wisdom and foresight. Until next time, keep questioning, keep exploring, and keep pushing the limits of what's possible.